In this lab activity, we're going to be looking at photosynthetic pigments. The goals for this lab: we want to discuss the purpose and function of photosynthetic pigments. We want to separate pigments from each other using paper chromatography. We're going to discern some of the chemical properties of these pigments based on their behavior during the paper chromatography. And we are also going to calculate the RF value for these pigments. So, what is a pigment? A pigment is a molecule that absorbs and reflects light. White light is made up of many different wavelengths, in fact, all possible colors of visible light. Pigments are molecules which absorb certain wavelengths of light and reflect others. The reflected light is the light that we see, so the reflected light is the light that was not absorbed. Photosynthetic pigments use the energy of the absorbed light to power the conversion of carbon dioxide and water into glucose and oxygen gas. Most plants have multiple photosynthetic pigments, and having multiple pigments allows plants to get energy from a wider range of wavelengths of light. Reflected wavelengths of light are of no use to the pigments reflecting those colors. When we look at a leaf and see the green color, that should tell us that those pigments are absorbing all of the colors except for green. Green light is not used in photosynthesis, but the other colors are. Paper chromatography is a technique that we can use to separate molecules from each other based on their chemical properties. So the pigments in plants may have slightly different chemical properties in addition to reflecting different wavelengths of light. One way to separate different chemicals is through paper chromatography. A chromatography solvent flows through a sample, and the components of that sample move at different rates depending on their solubility in the solvent and their attraction to the cellulose of the filter paper. So for this activity, for this protocol, we're going to start with a strip of filter paper. Now we want to measure to make sure that strip of filter paper is no longer than 9 centimeters or 90 millimeters. And that's because the chromatography chambers that we'll be using only support strips of filter paper about that long. Once you have your strip of filter paper, you want to measure 1.5 centimeters or 15 millimeters from one end, and then draw a line using a graphite pencil. This will be called the starting line, and our future measurements will all take place from this starting line. Our next step is to taper the filter paper. We're going to cut the end of the filter paper into a point where the widest part of that point ends up being where that starting line is up through the rest of the length of the filter paper. So you're going to trim the two corners of that strip of filter paper so we have a pointed piece of filter paper in which the widest part of the filter paper is where our starting line is present. We are then going to take a spinach leaf and a ridged coin or some smashing tool, and we're going to crush that spinach leaf along the pencil line. This is going to be the pigment sample that we will be separating. Now clearly that spinach sample is going to be a bit wider than a pencil line, but we want to make sure that our pencil line is within the area of smashed pigments. We are then going to take one of our chromatography chambers, which has a chromatography solution within it, and we're going to take our strip of filter paper, we're going to remove the lid, and gently put our filter paper within the chromatography chamber, quickly sealing the lid after we are finished. The chromatography solution within the chamber is very volatile, meaning it will evaporate very quickly. You might even notice it as a strong smell, and that's why we want to reduce the amount of time that the cap is off that chromatography chamber. This process is going to take about 7 to 10 minutes, but here we'll see an animation of what we would expect. As the chromatography solution moves up the strip of filter paper, we're going to notice pigments that had been all together down at the bottom will start to separate from each other. This is our paper chromatography experiment, and these pigments have separated from each other because of their chemical properties. Now, as soon as the solvent front or the liquid gets to about 1 centimeter from the end of the strip of filter paper, we want to stop this reaction and pull the strip out of the chromatography chamber. We quickly want to use either a pencil or a pen and mark how far the solvent front had traveled. that distance between the starting line and how far the solvent front traveled will be very important, and we'll use it in calculating a variety of other values. For each of the pigments, and we see 4 pigments that separated from that green stain down at the bottom, those 4 pigments, the 2 yellow pigments, a brighter green pigment, and a dark green pigment, we want to calculate the RF value for each of them. The RF value is the distance that the pigment traveled divided by the distance that the solvent traveled. And these measurements will all be taking place from that starting line down at the bottom. The RF value will always be between 1 and 0 because the pigment cannot travel farther than the solvent did, but the pigment will travel some distance from their initial starting point. I would recommend measuring from the middle of each of the pigment bands from the starting line. And so the distance that the farthest yellow pigment traveled would be the center of its band, whereas the distance the yellow pigment traveled, or the second yellow pigment traveled, would be about in the middle. The bright green pigment traveled so far, and the dark green pigment traveled a certain distance. For each of these, the distance the solvent traveled is the same, but the distance that the pigment traveled is different, and so we'll have a different RF value for each. You're going to be using a ruler to measure the RF value. Now, a few words about the RF value. It turns out that the RF value is a measurement of the chemical properties of that pigment. It actually has nothing to do with the pigment's color or which wavelengths it absorbed. Instead, it has to do with the pigment's solubility in our chromatography solution, and also its attraction to the cellulose of the filter paper. As long as the same solvent and same type of filter paper are used, the RF value is a constant, even if the filter paper was twice as long. If we allowed this to run twice as long as we did here, the RF value would still be the same because that ratio between how far the pigment traveled versus how far the solvent traveled is a constant. For this lab activity, when you make your measurements, please make sure you are measuring using millimeters. And recall, there are 10 millimeters in 1 centimeter. That's all for this activity. If there are any questions, please do not hesitate to ask.